Hello again. So I am your instructor until the end of this course. So it's the second half of the course. Okay, so we are having first lecture after your fall break, and it is about staining mitochondrial DNA, mitochondria themselves. And we're going to use a special tracker. So 10 people, you are 14. Okay, so I guess Orhan Basar, are you here? We're going to take your attendance anyway. So we are starting right now. Okay, so first of all, let's talk about the mitochondria. So what are they? And as you all know from the previous courses and from your school, they are the factory of energy. So what they do, they produce ATP after the degradation of sugar or simple molecules. Okay, so what, sorry, what is the source of these mitochondria in our eukaryotic cell? It is thought that at some point in the evolution of our eukaryotic cells, we actually tried to digest one type of prokaryotes, but we couldn't. And it actually led to the creation of these mutual beneficial interaction. So one type of prokaryotes, they became the energy factories for our cells. And since it was beneficial in terms of survival, etc., the cells which actually contained the nuclei, they were kind of pushed by the evolution to have the mitochondria too. Okay, so what we have these mitochondria, and their sizes, they range from 0.5 to 10 micrometers. So they are of a size of a usual bacterium, and they are found in our cytoplasm. So usually they have prolonged oval or roundish shapes. And it really depends from the cell type to cell type. For example, if you take the mouse stem cell, they would have spherical mitochondria. And it is the characteristic of this type of the cell. However, in our organisms, I mean the human body, they are usually prolonged oval-shaped structures. Okay, so as usual prokaryotes, they have the outer membrane and the inner membrane. Between these two membranes, we have the electric potential, which means that they have different charges in terms of electric charge. So one side is positive, another side is negative. They participate in the transmission of electron. And of course, in the end, they, because of this transmission, they produce lots of ATP molecules. And these ATP molecules, they will be used by the cell in the daily life and, for example, protein production or functioning of our cells. Of course, the cells, they differ depending on the spending of this energy. So let's say the muscles, because they contract, they require lots of this energy. And of course, they have thousands of these mitochondria in their cytoplasm. If you take liver cells, because they detoxicate the blood, they also need a lot of energy to actually remove the toxic part of the molecule. And they also require lots of energy and thousands of mitochondria found in the cytoplasm. What else? Okay, so neurons. Neurons, because they constantly need this change of the potential on their membrane. They also need to work against the gradient of concentration, which means that they need to spend a lot of energy. And of course, neurons, they have lots of these mitochondria too, while we have erythrocytes, and erythrocytes, they actually lack all the mitochondria because they need all the space which is available for keeping the hemoglobin. Okay, so what else do we know about the mitochondria? Is that they have their own mitochondrial DNA. So this DNA is coding for 37 genes, and it is made actually to make sure that the structures inside the eukaryotic cell, they don't become super independent. Otherwise, when we have these highly independent structures, they become parasites, which means that they start consuming the nutrients of the host, killing it. Okay, so it was not beneficial for our species. That's why some of the genes of these mitochondria, they were dragged into the nucleus of the eukaryotic cell. Okay, so this is about mitochondrial DNA and this type, this point here is that they divide by the binary fission as all prokaryotic cells. So they retain this type of reproduction because they are actually the prokaryotes. I mean, it is the most prevalent and best way to duplicate for small molecules that do not have a huge nucleus, for example. Okay, so what do we have? For example, on this image, on these two images, we have the muscle cells, and you could see the muscle fibers or sarcomeres. So one sarcomere is this small cell here. It's not a cell; it's actually a part of the fiber that participates in the contraction. Actually, sarcomere is from here to here. So this part is included in one sarcomere. Okay, so the mitochondria, which are stained green on this image, they are located between the muscle fibers. Why it's needed? Because these muscle fibers, because of this contraction, and this contraction is a very common function of these fibers, they will require lots of energy in situ. In situ means in the same place. That's why the localization of the mitochondria should be very close. And if you take the myopathies, so myopathy or myopathologies, some of these pathological conditions, they are characterized by the absence of these mitochondria between the fibers, which means these fibers cannot perform their function in whole or at all. Okay, so here you also see the muscle fiber and lots of these mitochondria stained green. So in this space between the fibers, we have huge number of the mitochondria, and the nuclei are stained blue here. Okay, so what are the main functions of the mitochondria in our organism? These are generation of energy, of course, in the form of ATP. And you all know that ATP contains the most energy, then ADP. And finally, we go to AMP. So monophosphate, diphosphate, and triphosphate. Okay, so as all types of work in our universe, any type of work includes the irradiation of heat. Why? Because the transition from one type of energy to another usually goes with the loss of some energy as heat. The same happens here, and here we have the transition from one type of molecule to another. So we have gradual degradation of sugar products into CO2 and H2O. So let's say we have H12O6. So we had this sugar here, and we want to degrade it to CO2 and water molecules. Of course, it includes lots of steps, and on every single step of that, we have loss of energy as heat. So they produce heat. Next, very important function is to store calcium. So calcium is the vital ion that is required in many cells for keeping the, for example, electrical charge, or to make sure that the contraction of muscle happens, etc., etc. So many stuff is actually related to calcium ions. And these calcium ions are stored inside the mitochondria. So if you are participating in the project of the research group that studies muscles, you will have some mitochondrial part of this project. Finally, the cell growth. Why cell growth? Because growth and division includes lots of energy spent. And of course, where do we get it? We get it from ATP and from mitochondria. But the thing here is that cell death is also related to our mitochondria. First of all, of course, when we don't have mitochondria in the eukaryotic cell, the cell dies because it lacks energy. Well, it's quite obvious, but the thing here is that on the membrane of these mitochondria, we have special proteins that sense the damage of the mitochondria. And if this damage is too huge or irreversible, then these proteins actually might initiate the apoptosis. And as you all know, the apoptosis is the programmed cell death. And if the content of the mitochondria does not get outside, and you might know that inside of it, we have radical oxygen species. We also have toxic enzymes. So if they get outside, then they start damaging the cells. So if we have too many of these toxic substances, then of course the cell could go into the necrosis. And as you all know, necrosis is not something that the organism wants to see inside. Usually we better go into the apoptosis than to necrosis. So apoptosis is the most prevalent or most favorable way of dying for the cell. So that's why we have these proteins that initiate apoptosis. So it's better to start apoptosis earlier than these toxic elements damage the cell and it goes into necrosis. Okay, so this is why the cell death is actually related to our mitochondria. Okay, so here you could see the mitochondrial diseases, and they could be related to the numbers. So we could have the increased number of mitochondria in the muscle cells. So you can see here that we have fibers with very few mitochondria, and a lot of them outside of these fibers below the membrane of this cell, which means that they are accumulated there, and most probably they don't work as they should. Also, they are of very strange shapes. and strange sizes, while the normal size that we usually have in our body is less than 3 to 4 sarcomeres. So sarcomere, as you remember, is this small unit here. So 3 to 4 are thought to be normal. Well, 3 is actually already long for mitochondria, but if we have longer, it means that we have problems with the size of them. Okay, so we might also have shapes. So let's say that the usual shape for this cell type is kind of prolonged oval, which means that we have structured crista, etc. And all of a sudden, this type of mitochondria becomes spherical, which means crista will not perform their function. And the function is to make sure that we have a huge surface for moving the electron, etc., for keeping the charge potential, etc. Of course, if we have the change of the shape into abnormal shape for this cell type, this is a huge problem for the cell. Again, about crista, they can be deficient. They can be stacking, which means we don't have a usual good shape of these crista. We have stacking. And stacking means that we disrupt the chain of electron transmission. It also could be hurled, like in storm. So you could see it here. Hurled crista also disrupt the transmission of the electron. Okay, they can have granules, which is not good. Just remember, if you have the mitochondrion, it should only have crista, nothing more than that. So the same goes for crystallic structures, and these crystallic structures, they can both damage the mitochondria or could be externalized, so they can accumulate outside of these mitochondria and damage the cell, which is also not good. So if we have this geometrical symmetry, it means that we might have some problems with these paracrystalline structures. Okay, so where again do we have these mitochondria, and what pathologies would be the result of the disrupted form or disrupted mitochondria? Of course, neural system, so epilepsy, the Alzheimer's disease, Parkinson's disease, etc., etc. So all kinds of problems with the brain because we cannot supply it with enough energy. The muscles, all the muscle pathologies, they might occur because of the very bad functioning of our mitochondria. And liver part that actually cannot detoxicate the blood, or, for example, the glands that produce hormones, because the production of the protein or any substance requires lots of energy. As you remember, the ribosomes, they use one ATP molecule to get one peptide bond. So it's disrupted if we have the deficiency in the ATP concentrations. Okay, so what we have here today during our lecture, during our lab, we have a special mitochondrial tracker, and this tracker is MitoTracker Red CMX ROS. So it comes from Invitrogen, and it comes as the powder. So it stains the mitochondria because it reacts with the charge potential or membranes that have the potential. Okay, so it is about the amine of this molecule. Why it happens? Because it has two functional groups, one which is recognizing the charge Another one is actually fluorophore. Just because it is a simple small molecule with a number of cyclic rings inside, it can get inside the cell without permeabilization. So we don't need the step of permeabilization to stain our mitochondria, which is really good because we could use it with the live cells. Okay, so when we are done with the staining of mitochondria, we can fix our cells with formaldehyde. And the good thing here, the formaldehyde does not reduce the signal that comes from our red CMX-ROS. Okay, so it retains in the cell after the fixation. And because we're using red color here, it is good for multiple staining of our cells. So we could use green, red, yellow, and blue for staining the structures of the cell. So we have excitation and emission range here. So it's given in nanometers, and it's shown here. As I said before, you're going to use red filter on your fluorescent microscope to image the cells. Okay, so now we are getting to our procedure. And first of all, we're talking about the materials and reagents. So we are using 96 well plate with NERC cell line. And we want to seed the cells beforehand so that at the time of staining, we have about 80% of confluency. So your well should be roughly occupied by cells about 80%. Then we're using the fluorescent microscopes, and all fluorescent microscopes in Nazarbayev University, they are research microscopes, which means they are very expensive. So when you're back on campus, when you have your offline laboratories and you are given task to go to take photos of your fluorescent cells, make sure that you have somebody who knows how to do that, because the cheapest objective, which is about 4x, is about 20K USD. And if we are talking about 100x objective, it goes 3-digit values. And if I'm not mistaken, it's about 200K. And of course, damaging these objectives not only puts the burden of the financial burden on the university, but also stalls the experiments of researchers in our university. So be careful when you are going to image the cells on fluorescent microscope. So we need the serum-free DMEM. Why do we need it? Because usage of usual complete DMEM, which includes the bovine serum, fetal bovine serum, it includes albumins. Albumins and lots of other molecules that actually can clump our fluorophores. So what we have, if we use the complete DMEM, we add our fluorophore, and one albumin can actually bind many of these fluorophore molecules, and they don't get inside of your cell. But it's not the worst part. The worst part is that this clump of albumin with the fluorophore can sit on your cell. So let's say this is your cell. And on top of it, we have these clumps of the albumins with the fluorophores. What happens when you image the cells? You have bright, very bright spots everywhere in your field of view, which means you messed up the whole experiment and you will need to redo it. And sometimes it's really crucial because you don't have enough cells, or you have cells that you are going to image in a very long-term project, which means you were preparing these cells for, let's say, 3 months, and then you just messed up the experiment by adding the fluorophore into the complete DMM, which means 3 months are wasted. So always, please keep your eyes on serum-free DMAM when you talk about staining inside of the medium. Okay, afterwards, when you stain the cells, you're going to add the complete medium. So you remove the fluorophore and add the complete medium. Why you need the complete medium? Because you want the cells to survive your experiment. Okay, so this is about the materials and reagents. Now we're talking about the tracker itself. So it comes in a vial as a powder. So you have 50 micrograms or 50 times 10 to the power of minus 6 grams. And you want to dissolve it in DMSO because you are going to use very small amounts of this stock solution. The DMSO is not going to damage your cells, so don't worry about this part. I hope you all know that DMSO is actually toxic, but in this case, it's okay because we are using very low concentrations. And the thing is, if we try to dissolve our tracker in something else, it will not happen because this tracker is actually hydrophobic. Okay, so what we do, we take this amount of the MITRE tracker, and we know its molecular weight, which is in grams per mole. Then we need to identify the amount of this substance. So we divide mass by molecular weight, and we get roughly 9.4. So it's 0.4 times 10 to the power of minus 9, or 9.4 times 10 to the power of minus 8 moles. Okay, so this is the amount of the substance, and now we are calculating the concentration. And as you remember, the concentration in terms of molar concentration, it is the amount of the substance divided by the volume. So we have this amount of substance divided by 94 microliters of the DMSO, and in the end we get 1 millimolar concentration, which is written here. So let me write it like this. So this is your stock solution, okay? You are going to keep it at minus 20, protected from light, because it's the fluorophore. And of course, we have the photo bleaching effect. We don't want it. That's why we keep it usually in aluminum foil. Okay, so when we are ready to stain, we need to get this stock solution, which is 1 millimolar concentration. We need to bring it to 10 micromolar concentration. So to this end, we are, as always, we are using the mother formula of LabMath, which is C1V1, C2V2. So in this case, C1 would be your 1 millimolar. V1 would be X, because you want to identify the volume that you need to take. C2 would be 10 micromolar in this case, so they are different. And V2 would be equal to 500 microliters. Okay, so after the calculations, you obtain 5 microliters of your stock solution. In total, 500 microliters of the end staining solution, final staining solution. And of course, it makes you 495 serum-free medium plus 5 microliters of your stock solution. Microliters. And you get 10 micromolar concentration. Okay, so Do you have questions here, guys? Do you even hear me?
Yes, we hear you.
Yes, we hear. No questions.
Okay, nice, no questions. Okay, so we have the procedure itself, so the protocol. First of all, of course, when we obtain our cells, we want to remove the medium because it contains the albumins, the waste products, which could also clump lots of fluorophores. So you want to remove everything from the well. And after you removed everything, you're going to add 100 microliters of the serum-free medium to the well, and then add 2 microliters of your staining solution. Okay, so you have again 2 microliters of your staining solution, which is 10 micromolar, and you obtain 100. Well, it's 102, but because it's not clear calculation, we could just round it to 100 microliters. So the X solution here would be 2 times 10 micromolar divided by 100. So these microliters, they will cancel each other, and we're left with this micromolar. And of course, 20 divided by 100 is 0.2 micromolar, or 200 nanomolar concentration. So this is where you get these 200 nanomolar concentration from. Okay, so when you added this staining solution, you are going to incubate it for 30 minutes in the incubator. Why? Because your cells are alive and you don't want to kill them at room temperature. So you keep them protected from light. Usually incubators, they don't have internal light, but there are different types of incubators. Some of them, they have actually glass shields, which means light comes easily inside, which means you need to protect your 96 well plate with some kind of foiling or covering of foil. Okay, so after 30 minutes, you're going to remove the staining solution. And instead of the staining solution, you're going to add complete medium, the same amount. And after that, you're good to image your cells. Of course, the 96 well plate that you're going to use, it has special bottom, which is glass. This glass is compatible with the fluorescent objectives of high resolution and high magnification. And usually they are very expensive. I don't say it to kind of scare you. I just say it to make sure that you understand the responsibilities that come with the project. Okay. So this is about the protocol itself. And the thing here is that we don't use high concentrations. Why don't we use it? Because if we use high concentrations, the dye, the fluorescent dye, it will bind to everything that has some kind of potential or has this membranous structure. So it could bind to lysosomes, it can bind to endoplasmic reticulum, to Golgi apparatus, to nuclear envelope, etc., etc. That's why we want to keep it as low as possible. So 200 nanomolar might be fine. At some cases, you might actually decrease it. So you don't decrease it just 10 times straight away. You're going to test it. So you decrease it 2 times, then 4 times, then 8 times, etc., etc. So you want to find the best staining condition in the way that you don't get a lot of the background fluorescence when you do your experiment. In this case, when we were doing the video shooting, etc., Aijan found that 200 was quite fine. So 200 goes in the standard protocol. So it was fine for the cells that we used in the video. Okay, so this is the image that we got. It is 4x magnification. We don't have higher magnifications because at that time point, when this image was taken, the computers connected to the microscopes, they had the virus that actually deleted much stuff. And that's why we have to operate with this not magnified images. But here you can see that actually we have nice staining of the cytoplasm. And as you understand, in the cancerous cell, although it has lots of anaerobic glycolysis, which means the degradation of glucose in anaerobic way, in this case, we obtain 2 ATP molecules. And it is not fine, but when the cancerous cell leaves, it cannot wait for mitochondria to produce the 38 ATPs in the transmission of the electron chain. It just straight goes into this anaerobic path of the degradation of glucose. That's why they have lots of byproducts that actually intoxicate cells. Still, in these cancerous cells, we have lots of mitochondria that might work abnormally, but still they are there. That's why we have this staining, and it is very good staining actually. Sometimes you don't have much signal, which means that you might want to increase your concentration. It might be related to the age of the tracker, which means it was kept for a long time and started degrading itself without photobleaching, which means that you might want to perform step of experiments to identify the best concentration that will stain your mitochondria, but will not stain other membranous organelles in your cell. Or you actually might want to totally waste this stock of your tracker and start new one. Okay, so here you could see a better, a higher magnification of the cells. So it is a confocal analysis of HeLa cells. So you could see the cytoplasm here. You could also see the nucleus. And they were, in this particular experiment, they were staining lamin. And lamin is the part of nuclear envelope. So nuclear envelope contains one particular, in this case, actually 2 particular proteins, and they were aiming it to. Here, you can see the red mitochondria, and they're spread evenly in the cytoplasm. So here, I would say that they kept dye more than it was needed, or they just used very high concentration of the fluorescent dye. The thing here is that it is tricky when we have green color. Why? Because our eye is much more comfortable in seeing green than red and blue. Why it happened? Because as apes or as primates, we were living in the very green conditions. So in forests, that's why it was better for us to see more shades of green than red and blue. That's why we kind of see better the green light. To decrease this kind of skewing in color seeing, we actually might take the monochrome images of these channels. And if we take the monochrome of this red channel and monochrome of this green, we would see the real values of the intensities. So black and white might actually seem the same. But for me now, when I look at green and red, I perceive it as better coloring here and overexposed here. or oversaturated. Well, again, the best would be seeing it in monochrome channel. Okay, so now we have.
Sorry,
two
can I ask
slides.
a question?
Yes.
So for the results section, what picture should we use as a reference?
I would go with this reference. I mean, here.
The red one, yeah?
Sorry.
And but these are not NRK cell line, yeah?
Yes, it's not NRK.
So should we write like that our results are for the HeLa cell line? But so what should we discuss?
Yes, just
HeLa
imagine,
or NRK?
Just state that you are going to use the reference images from HeLa. Well, let's do it like this. I'm not really sure if this particular lab is actually in your lab book or lab report. So if it is, I will try to find the NERC images with mitochondria stained and will send them to you. If I don't, just take this particular image and write it in your lab book that you actually had only HeLa cells stained with red mitotracker, okay?
Okay, thanks. I think we have the same situation with tubulin and host, or how it's pronounced. Let's say hext. This one. Okay,
yeah.
Hex. Yeah. Also, HeLa cell lines were provided in the presentation. So can you please send us the correct images or just write?
Okay, I will send the NERC images of the tubulin staining. They are of a very good quality because they are confocal too. So you won't see them on usual fluorescent microscope, but you could draw it as the image of the previous lab. I'm pretty sure it should be fine.
Okay.
Nice. So we have 2 slides of questions. And do we have volunteers here who would want to take either of these slides, or should I assign people? Okay, so first slide is Ayjan Talibai.
Yes, I'm here.
Nice. So we are talking about the stock solution here. So go ahead and solve the problem.
Okay, so MitoTracker dye stock solution was prepared for 94 microliter DMCO, was added to the vial content, 50 micrograms, to achieve how much solution. So, and the molecular weight is written here.
Yes, so how do you start?
So should we use the formula, the basic formula, where we like volume on concentration?
Choose whichever.
Honestly, I don't know how to solve this problem.
Well, it's like 15 minutes ago, I already shown how to solve it. Do you remember the formula? Ayjan?
No, sorry.
Any glimpse of formula?
No, sorry.
Okay, so we have Aygirin Koshurova.
So we should divide 50 microgram to what's the molar weight, and then it will be the moles. And then we should divide it by the volume to obtain the concentration.
Okay, nice. So the concentration is equal to amount of the substance divided by volume, while the amount of substance is equal to mass divided by molecular weight. Okay, so go ahead and solve it.
So 531 is in gram, and we have the mass in microgram. So we should convert it into, it will be 0.05 grams divided by this 500. It will be
sorry, it's not 0.05 grams. It's gonna be.
I'm really
50.
sorry, Ali.
Yeah, it's.
We are going to talk to you after. Okay.
Okay.
Now Aigerim is answering. Aigerim, we are still talking to you.
So will it be? Isn't it not? Is it in microgram?
It is microgram. Yes.
Oh, sorry. Then it will be 0.094 moles.
How do you get it here?
I just divided 50 on 531.
Okay, Aigerim, the problem here in your answer is these micrograms. Micro means 1 millionth. Okay. This, what you gave us, is for 1/1000th. It's not the same. Okay, Ali, it's your turn.
Okay, so it's gonna be 50 times 10 to the power of -6.
50 times 10 to the power of -6. Yes. So you convert this microgram to gram.
Grams,
And
yeah,
it
and
becomes-
then delete it. Not delete, but divide it by 531.5. I don't have calculator.
Anyway, so what is the measure unit here?
It's gonna be grams.
Grams, nice. So who has the calculator then? Anyone?
It's actually easier to calculate the grams per liter concentration first and then convert. Then you will not basically need a calculator.
It's the same actually. Paulina, okay, so we're talking to you now. So
I
do you
just
have
took-
any
I just
problems? Do you have any
just
problems
took-
with this one?
Not with this. I just first divided 50 micrograms by 94 microliters. So micro, they just cancel, and I only
Okay.
have grams per liter left. And then I
So how much
divided
is
by
that?
that will be so the same numbers like 0.5315236. So like
Okay, so
almost like in the molecular weight,
Yes.
approximately.
So what is the
So-
measure unit?
Of the formula that you've written.
No, for the formula you said before, like dividing this part by this
So
part.
that will be just grams per liter.
Grams per liter. Okay, so what is the next step then?
Then I divided it by the molecular weight.
Okay, so tell me the molecular weight.
Well, here it's you wrote grams. I assume that was grams per mole.
Of course. So it is gram per mole because without per mole, it is not the molecular weight. It is mass. We're not talking about mass here. That's why we need this gram per mole measure unit. Okay, so we divide it by 3152 grams per mole.
So that will
What
be
happens here?
approximately 1 millimole.
Of course. But the thing here is we can cancel grams. If we don't have mole after that, I mean, in the divisor, then we will have something divided by liter. And of course, concentration is never something divided by liter. That's why this mole goes up, and we're left with 0.001 mole per liter, or 1 millimolar concentration. This is absolutely right. It's just the other way of taking the same calculations. So if we go this way, we would still get 94 times 10 to the power of something in moles. Okay, okay, so let's do it right now. So here it's -7, 94 minus 7, 9.4, like this. Okay, so it's 9.4 times 10 to the power of -8 moles. Now you're going to divide it by 94 times 10 to the power of -6, and you're left with 1 millimolar concentration. Okay, so it's 9.4 times -9 should be. Okay, so it's the same way as Paulina said, but taking this part first and then going to this part. So what Paulina did, she got mass divided by volume and then divided by the molecular weight. So the same, you either take this part first and then this part, or vice versa. So it's the same, guys. So just don't get scared by the formula, etc. The calculations is the common part of your research. So whenever you get to laboratory, expect to have lots of math, specific type of math, lab math, and you have few formula to remember. Now you only need to combine them. So the same way you calculate concentration like this, you can calculate the concentration from the diluted solution. So if you have two solutions, so let's say this is stock and this is your staining solution. So C1, V1, C2, V2. So the thing here is that the same amount of substance that you take from stock solution goes to your staining solution, which means this N here is equal to this N here. And in this case, N would be equal to, if we move this V1 to the other side, it will be equal to C1, V1. And N for this part would be equal to C2, V2. Since they both are the same, then you can equalize these parts too. C1, V1 equals to C2, V2, okay? So this is very simple. You have to know very few formula, and this very few formula will give you the superpower in your research, okay? If you know how to do the calculations, it means that you do not mess up. If you do not make mistakes in your laboratory practice, then you are a very valuable member of the group. If you constantly do the mistakes, it means that you will bring only spending to the lab, which is of course unfavorable. And people will talk about it. If you do not change your way of doing stuff, of course, you will not progress in your scientific career, okay? So this is very simple. And because of its simplicity, people underestimate it, they skip it, and then they make lots of mistakes. That's why, please learn how to calculate in the lab. Of course, again, this is not quantum mechanics, where the formula include like differential math, etc. So all these scary part of the mathematics, it is very simple. It is not even the whole chemistry math. It's a very small part of it. That's why, please, I will send some videos to you so that you kind of get familiar with all these calculations. But please do practice yourself too, because without it, you will not be able to play a valuable role in the laboratory. Okay, so all these small tasks, they prepare you for the real lab work. Okay, so we have the second question here.
Can I try to
And-
solve it, please?
Ayjan? Yes, of course.
So it's the same formula that you showed us at the end. Can I write on the screen?
Of course.
Mm.
Just tell me what to do. I'm going to write.
Can I write by myself like this?
Oh, nice. I didn't know you could do that. Okay.
Like C1 V1 equals to C2 V2. But before multiplying, I have to make units the same. So I have to make
Just a
this
second,
one.
Ayjan. Do you guys see it too?
Yes.
Oh,
Yes,
nice.
we see it, yes.
I didn't know
I have
that
to-
you could do that.
Magic.
Okay.
Okay, so I have to convert these units so that they will be similar. So
Okay.
in one, it's 1,000 of microliters. Sorry.
It's okay.
And now I have to multiply 10 micro to 500, so it will be like 5,000. Equals to this concentration multiplied by the volume, V2, which will be equal to 5, but it's so it's the only answer, 5 microliters. So I need to obtain 5 microliters from the stock and add it to the PBS. So in order to find the PBS, I have to just 500 microliters. Subtract, and it will be 495.
Absolutely
correct?
right. Yes, absolutely right. So for those who didn't understand, this part is 10 micromolar times 500 microliter. So this microliter is your V1 and this is your C1. You could also name them C2, V2, it doesn't matter. So you just have to keep the same or correlated values on the same side. So another part here, so it's not microliters, it's micromolar. So 1 millimolar is equal to 100 micromolar. Okay, so everything else is super fine. So we equalize these two parts. And now we have V2, which is division of this part. And of course, here we have micromolar times microliter divided by these 1,000 micromolar. Of course, these micromolars, they cancel each other, and we are left with microliters only. That's why here we have 5 microliters. Finally, because we know the final volume, it means that some part of it is from stock solution, another part is from PBS. So what you do, you have this 500 microliter minus your found value, and you're left with 495 microliters. Absolutely right. Okay, so we have done these two parts. Now we are getting to this slide. Okay, so who wants to do this? Ayjan, I'm not really sure how to delete it because I didn't know how to do it at all. So could you please remove them? Okay, so I don't know how to do that. Okay, annotation, right?
I think you can delete it all at once by pressing "Стереть все" button. It should be in the middle upper part of your screen.
Yeah, that's all I deleted.
Okay, nice. So I see now, sorry. Yeah, so there is an eraser and clear all. Okay, cool. Now I know another cool feature of this program. Okay, so who wants to do this part? Not people who already talked. So let's go with Dana.
I'm sorry, what we need to match this or what?
Yes, so it's a
Okay,
match.
now I see what the problem.
Okay, so Dana, are you here or do I?
Yeah, I'm here.
Okay, nice. So you have first two answers.
Okay, homogeneous mixture, it's a solution. And
Why?
then, well, because one compound dissolves in the liquid compound, and so
Yes,
it
absolutely
creates
right.
homogeneous
So
mixture.
they are not chemically bound. This is the main part here, which means that one player here is dissolvent, another part is solvent and solid. And
Yeah.
these are characteristics of the solution. Okay, so second part.
And what measures amount of mass per unit of volume is solution density. Like,
Why not concentration?
because concentration measures amount, like in moles per volume. And
No,
in
it's
case-
not always the same. So it could be in grams per liter.
Oh,
So what makes you think that it is the density, not the concentration?
I have some.
Okay, actually it comes from this from the third part. So go ahead with the third one.
Okay, it describes amount of the compound dissolved in the solution. So the difference here is, so yeah, in the case of density, we mean the whole solution as a whole per unit of volume, and in case of concentration, we mean the amount of solute in the solvent.
Yes, so the main word here is dissolved, which of course gets you to the question of solution. And they can be in the same measure units, but for the sake of this particular task, of course, this part is related to solution concentration, while this part is related to the solution density. Okay, so what is the last one then?
Increase of solution concentration results in
Well, we
increase
all
of its
understand
density.
here.
Yeah.
Increase or decrease?
Increase.
Why?
Usually the solid substance that is being dissolved has higher density. So if we add more of this solid substance, then it increases density of the solution too.
Yes, so here you would talk about the correlation. So you have N per volume related to mass per volume. Why? Because N is the mass divided by molecular weight. So with the increase of mass, with the increase of amount, we also have the increase of mass because the molecular weight is the same, right? Because it's the same density and the concentration of the same solid. Because they are related through this formula, we have this ratio. So it's straight. I'm not really sure how to call it. So it's not reverse, it's straight ratio. With the increase of amount of compound, we increase the mass and vice versa. Okay, so this is right, Dana. And actually it is the end of today's lecture. Now we switch to questions.
I'm sorry, I have a question.
Of course.
But it is more about the lab book submission. It is written on the Moodle that we have to submit it tomorrow on 16th, but in the syllabus it is 17th. So will you change it?
Yes, I will.
Okay.
The thing is, it's going to be 17 hours, so it's 5 o'clock. It's not going to be
so
midnight.
it's not midnight.
Okay. So in the syllabus, it was written midnight, but we decided to keep it 5 o'clock.
But the date is 17th.
Yes, I'm going to change it right now.
Thank you.
You're welcome. Okay, so any other questions, guys?
So again, for the experiments of the lab notebook, will you send the results, or can we use those that are in the presentations?
Okay, again, if it is going to be the experiment for lab book submission or lab report submission, of course, I will try to find NERC cells stained with MITRE tracker. If I don't find it, you could use the HeLa cells given in the slide, and Orunbasaro, of course, will accept it because it's the unfortunate case of computer virus on our microscope computers. So I will update you on that, so don't worry about it. The next week should be your midterm, which is also I'm going to talk about with other instructors, because it should not be the same day as your lectures. I will update you again about it. So any other questions?
Can I ask about midterm? Will it be on Moodle?
It's going to be on Moodle. So
Oh,
on Moodle, we have the trackers. So they track your interaction with the computer. So they will they will log everything that you do without saying what exactly you do. So if you open, let's say, the PowerPoint program, it will tell us that you are having PowerPoint application running on the background. It will not tell us what exact lecture you are looking for. If it is lecture at all, you might be looking, let's say, cartoons. It doesn't care. Or it will also log the tabs in your browser. So it tells us like the student with the identification number this and this is using other tabs on in the browser. It does not tell us if it's YouTube, if it's your email or anything else, but it tells us that it's something else than your Moodle exam. And of course, we leave the right to actually cancel your mark for midterm if we find out that you were using lots of side tabs or programs like Acrobat Reader, PowerPoint, etc., because it is not open book exam. Okay. So any other questions?
I actually have a question about Lobster.
Okay.
So like I try out some of it, and I noticed that when it's like the part of spectrophotometers, when I did it, like it stuck and would not proceed any further when it's time to calibrate the graph. So like what, I don't know to whom I should, like I wrote to their help center, but nothing happened.
Could you please send an email to me with the explanation of the problem?
Okay.
So that I can address it to Lobster help desk, because they are actually waiting for our feedback. And of course, if you have anything to share, it would be great.
Okay, I will.
Nice, thank you. Okay, any other unclear parts that we should talk about? Nice. So let's call it the end of the lecture. I will see you next week. Also, you will wait for the updates from me about the results section and the midterm. I hope that we have a very smooth and enjoyable continuation of the course. So I will see you next week. Goodbye, guys.
Okay, bye.
Goodbye.
Thank you.
Bye.
Goodbye.